Hello viewers, welcome to my channel. In continuation of the previous video, today's video we will have the question answer session on microbial growth, nutrition and metabolism. So let's start. The first question is, a bacterium that uses reduced one carbon compound such as methane or methanol as its sole source of carbon and energy, known as option 1 Methanotroph, option 2 Methylotroph, option 3 Methanogenes, option 4 Mixotrophs, and the correct option is Methylotroph. In our previous video, we will already discussed about the Mixotroph. Methanotrophs are the microorganisms with the ability to grow on methane as the sole source of carbon, whereas Methanogenes are microorganisms. These are strictly anaerobic archaea that obtain energy by converting carbon dioxide, hydrogen, acetate, formate and other compounds to either methane or methane and carbon dioxide. The next question. The association in which the growth of one organism either depends on or is improved by the provision of one or more growth factors or nutrients by neighboring organism. Sometimes both organisms benefit. This is known as option 1 Syntrophism, option 2 Mixotrophs, option 3 cometabolism, option 4 Methanotroph. And the correct option is Syntrophism. In our previous video and earlier slide, we have already discussed about the Mixotroph and Methanotroph. So in this slide, we will discuss about the cometabolism. So often it is found that the addition of easily metabolized organic matter such as glucose increases the biodegradation of recalcitrant compounds that are not used as carbon and energy sources by microorganisms. This process termed as cometabolism and can be carried out by simply adding easily catabolized organic matter such as glucose or cellulose and the compound to be degraded to a complex microbial community. Next question. The vitamin required for Lactobacillus species is option 1 folic acid, option 2 niacin, option 3 pyridoxine, option 4 riboflavin. And the correct option is niacin. Niacin is a form of vitamin B3. It is also known as nicotinic acid. It can be produced from the amino acid tryptophan by plants and animals. Whereas folic acid is vitamin B9, pyridoxine is vitamin B6 and riboflavin is vitamin B12. Next question. Nitrites are oxidized to nitrate by which microorganisms? Option 1 Nitrosomonas, option 2 Nitrosococcus, option 3 Nitrobacter, option 4 Azotobacter. And the correct option is Nitrobacter. Ammonium is converted to nitrite by Nitrosomonas and nitrites are oxidized to nitrate by Nitrobacter. This process is known as nitrification. Next question. Microbial population enters stationary phase for option 1 accumulation of toxic substances, option 2 depletion of nutrients, option 3 reaching a critical population, option 4 all of this. And the correct option is all of this. In stationary phase, the growth rate becomes equal to its death rate. The cell population remains constant at this stage. If a graph is plotted with this growth phase, a smooth horizontal line will appear. Next question. The media that supports the growth of a particular microorganism are known as option 1 differential media, option 2 selective media, option 3 supportive media, option 4 enrichment media. And the correct option is selective media. Differential media allows the differentiation of different groups of microorganisms and even identify the microbes based on their biological characteristic. Example, blood agar. Enrichment media. This media which encourages the growth of fastidious microbes. Blood and other special nutrients are added to encourage the growth of fastidious microbes. This specially fortified media, that is blood agar, are called as enrichment media. Whereas supportive media is a media such as tryptic soy broth, and tryptic soy agar are called general purpose media or supportive media because they sustain the growth of many microorganisms. The next question. Which of the following types of media would not be used to culture aerobes? Option 1. Selective media. Option 2. Reducing media. Option 3. Differential media. Option 4. Enrichment media. And the correct option is reducing media. A reducing media is a media where reducing agent such as sodium thioglycolate is added to remove oxygen. Reducing agent chemically combines with oxygen dissolved in the medium and makes it unavailable for the microorganism. It is used to culture anaerobes. Next question. The major constituent in agar are option 1 fat, option 2 amino acid, option 3 polysaccharide, option 4 polypeptide. And the correct option is option 3 polysaccharides. Agar is totally inert with no nutritive value. Most microorganisms are unable to degrade it. Agar melts at 85 degrees centigrade and solidifies between 32 degrees centigrade to 40 degrees centigrade. Agar is originally derived from red algae. Next question. McConkey agar medium is a option 1 enrichment medium, option 2 selective medium, option 3 differential medium, option 4 both 1 and 2, option 5 both 2 and 3. And the correct option is both 2 and 3. That is McConkey agar medium is a selective medium as well as differential medium. The selective components in MAC agar are bile salts and crystal violet, which inhibit the growth of gram-positive bacteria. The pH indicator, that is lactose and neutral red, distinguish the gram-negative bacteria from the gram-positive bacteria based on the products released when they use lactose as a carbon and energy source. The colonies of those that release acidic products are red. Example is E. coli. Next question. The compound that is added to the medium to absorb oxygen for creation of anaerobic condition is option 1 sodium thioglycolate, option 2 nitrous acid, option 3 citrate, option 4 none of these. And the correct option is sodium thioglycolate. Sodium thioglycolate is a reducing agent which is added to the media that would remove oxygen and makes the media reduced. Reducing agent chemically combines with the oxygen dissolved in the medium and makes it unavailable for the microorganism. The next question. The mean growth rate constant expressed as option 1 K is equal to log NT minus log N0 by log 2. Option 2 K is equal to log NT minus log N0 by 0.301 into T. Option 3 K is equal to log N0 minus log NT by log 2. Option 4 none of these. And the correct option is K is equal to log NT minus log N0 by 0.301 into T, where the N0 is the initial number of population of bacteria, NT the population of bacteria at time T. The growth rate constant. The exponential growth rate constant can be described with the mean growth rate constant, that is K. The number of generation in per unit time T is regarded as the mean growth rate constant. In case of microorganism, it is usually expressed as generation per hour. The next question. The number of generation in time T expressed as option 1 small n is equal to log NT minus log N0 by log 2. Option 2 small n is equal to log NT minus log N0 by 0.301 into T. Option 3 n is equal to log N0 minus log NT by log 2. Option 4 none of these. And the correct option is n is equal to log NT minus log N0 by log 2. Where N0 the initial number of population of bacteria, NT the number of population of bacteria at time T, N the number of generation in time T. Thank you. Please like, share and subscribe to this channel.